How can we uncover the hidden secrets of a molecule's identity and structure with just a tiny sample? Mass spectrometry, often referred to as the molecular detective, achieves this by precisely measuring the mass-to-charge ratio of ions. The process begins with a sample that is first vaporized into a gas phase. Once in this state, the molecules are ionized, often by bombarding them with high-energy electrons or using lasers to create charged particles. These ions are then accelerated into a mass analyzer, where they encounter an electric or magnetic field that separates them based on their MZ ratio. Lighter ions move faster and travel farther, while heavier ones lag behind. The result is a mass spectrum, a graph showing peaks that correspond to the masses of the molecule's fragments. For example, analyzing water might produce peaks corresponding to hydrogen, with a mass of 1, and oxygen, with a mass of 16. Scientists can piece together the information from these peaks to determine the molecular structure and composition. This technique is indispensable in fields ranging from drug discovery, where it identifies pharmaceutical compounds with incredible precision, to environmental science, detecting trace pollutants in air and water. Whether solving crimes or exploring space samples, mass spectrometry opens a window into the invisible molecular world.
Mass spectrometry, MS, is an analytical technique used to measure the mass-to-charge ratio, MZ, of ions. By analyzing these ratios, MS can identify and quantify molecules, determine their structure, and study their chemical properties. The technique relies on three main steps: ionization, mass analysis, and detection. Mass spectrometry is widely used in fields like chemistry, biology, environmental science, and forensics due to its sensitivity and precision. It can analyze a wide range of substances, from simple gases to complex biomolecules like proteins and DNA. Mass spectrometry is used to detect drugs in forensic investigations by identifying specific molecular fingerprints. Mass spectrometry is like a molecular detective, uncovering the identity of substances by analyzing their atomic fingerprints. The first step in mass spectrometry is converting neutral molecules into charged particles, ions. This is achieved through ionization techniques such as electron ionization, EI. High-energy electrons knock electrons off molecules, creating positively charged ions. Electrospray ionization, ESI. A fine spray of charged droplets produces ions, ideal for large biomolecules. Matrix-assisted laser desorption ionization, MALDI. A laser vaporizes a matrix containing the sample, ionizing it for analysis. Ionization is crucial because only charged particles can be manipulated by the electric and magnetic fields used in mass spectrometry. In proteomics, electrospray ionization is commonly used to ionize proteins for analysis without breaking them apart. Ionization is like giving molecules an electric charge so they can be guided through the mass spectrometer like tiny magnets. Once ionized, the particles are directed into a mass analyzer, where they are separated based on their mass-to-charge ratio, MZ. Common types of mass analyzers include quadrupole, uses oscillating electric fields to filter ions of specific M per Z values. Time of flight, TOF, measures the time ions take to travel a fixed distance, with lighter ions arriving first. Ion trap, traps ions in a stable orbit using electric fields and sequentially ejects them for detection. The mass analyzer is the heart of the mass spectrometer, enabling precise measurement of ion masses and providing detailed molecular information. Time of flight mass spectrometry is used to analyze complex mixtures like crude oil, separating thousands of components based on their MZ. The mass analyzer is like a racetrack for ions, sorting them by weight as they cross the finish line. After separation, ions reach the detector, where their signals are recorded and converted into a spectrum. The detector measures the abundance of ions for each MZ value, creating a mass spectrum, a graph showing peaks that correspond to different molecules or fragments. Modern detectors, such as electron multipliers or Faraday cups, are highly sensitive and capable of detecting even trace amounts of substances. In environmental analysis, mass spectrometry can detect trace levels of pollutants like pesticides in water samples. The detector is like a microphone, capturing the voices of ions and translating them into data. The mass spectrum is a visual representation of the data collected during analysis. Peaks on the spectrum indicate the MZ values and relative abundance of ions. Each peak corresponds to a specific fragment or molecule, enabling identification and structural analysis. Advanced software is often used to deconvolute complex spectra, making it easier to identify compounds and quantify their concentrations. A mass spectrum of a drug shows a peak corresponding to its molecular ion, allowing researchers to confirm its identity. Interpreting a mass spectrum is like decoding a bar graph to understand the composition of a puzzle. Mass spectrometry is a cornerstone of proteomics, protein study, and genomics, DNA and RNA analysis. Techniques like tandem mass spectrometry, MS, allow the fragmentation of ions, providing detailed information about molecular structures and sequences. In proteomics, MS identifies post-translational modifications and protein interactions. In genomics, it helps study nucleic acid modifications and identify biomarkers for diseases. Mass spectrometry identified biomarkers for early-stage Alzheimer's disease by analyzing protein modifications in blood samples. In proteomics, mass spectrometry is like piecing together a jigsaw puzzle of proteins to reveal a biological picture. Forensic scientists use mass spectrometry to analyze evidence like drugs, explosives, and trace chemicals. Its high sensitivity allows the detection of substances even in minute quantities, making it invaluable for criminal investigations. Coupled with gas chromatography, GCMS, it separates and identifies complex mixtures, such as detecting illicit drugs in body fluids or analyzing residue from explosive materials. Example: mass spectrometry detected unique chemical signatures on bomb fragments, aiding investigators in identifying their origin. Analogy: mass spectrometry in forensics is like Sherlock Holmes's magnifying glass, uncovering hidden clues from tiny traces. Mass spectrometry plays a critical role in monitoring environmental pollutants and ensuring food safety. It detects contaminants like pesticides, heavy metals, and toxins in water, air, and food samples. With its ability to measure parts per billion or even parts per trillion concentrations, MS ensures compliance with safety standards and protects public health. Example: mass spectrometry identified mercury contamination in fish, helping authorities issue consumption advisories. Analogy: mass spectrometry is like a health inspector for the environment and food, spotting dangers invisible to the naked eye. Ongoing advancements in mass spectrometry are enhancing its speed, sensitivity, and accuracy. Innovations like ambient ionization techniques enable real-time analysis of samples without complex preparation, while miniaturized devices bring mass spectrometry to field applications. Combining MS with artificial intelligence and machine learning is also revolutionizing data analysis, enabling faster identification of complex compounds and patterns. Example: portable mass spectrometers are being used in space missions to analyze the composition of planetary surfaces. Analogy: future innovations in mass spectrometry are like upgrading a camera to capture finer details, faster, and in more challenging environments.